right, there we are. We are back. At Christmas, we have we have decorated. What's up? What's up? Moped Vic, Noomba, Space Camp, hello. Kelsey, hello, welcome in. Molly, granola, welcome back. Tortilla, Nikki, what's up? So, tonight the plan is just uh get back into Beetle ID. Again, we have moved the Wednesday streams to the science category rather than just chatting. Ooh, I never remember to close all of it. Um, and yeah, so we're just gonna get started with our first and probably the most frustrating of the beetle families. Space Camp, I think you're fine. I I don't really know how different the demographic is, but um, I was staying and just chatting for most of the things because we do tend to yap quite a bit. Um, and I was saying the one time on stream, I was like, I feel like in the science channel, all the streams that I've watched in there, they have like a lesson plan almost. And someone very wisely pointed out, well, Pre-Chatchats has a lesson. So we're trying this. We're gonna see how it goes, and maybe get new members. Who knows? Okay, so to reorient where we are in the whole big grand scheme of things again, um, we are in the suborder Polyphaga, which is the largest within beetles. So there are like 160 some families of beetles worldwide, and 140 or so of them are in Polyphaga. So this is the major radiation, and this is where the highest diversity of our beetle species are. And within Polyphaga, we are inside of the Tenebrianoidea, which is the superfamily of things that are related to this guy right here. You have seen him several times. This is a darkling beetle, one of the desert darkling beetles. And so everything in this subfamily, or superfamily, excuse me, has some physical characteristics that kind of relate back to this guy. The big one that we have talked about, and I know has been frustrating, has been the tarsal counts. So their tarsal formula in this superfamily is 554, which means that on the front and middle legs there are 5 segments, and on the hind legs there are only 4. So this is a front leg right now, and you have 1, 2, 3, 4, 5 segments, and then the last segment has those claws on it. The claws are not a separate segment, they are just simply attached to the last tarsal segment. And the insect leg as a whole is also divided into 5 segments. So closest to the body where it attaches is called the coxa. And then there is a trochanter, a femur, a tibia, and then the tarsus. So the femur and the tibia are the longest sections of the legs. And then the tarsi combined are basically like the foot. They're the part that can articulate and has the claws to grab on and, uh, hold on to things. This is the front leg. So again, 5 segments. Now the middle leg, also hopefully a pretty easy, pretty obvious 5 segments. First one's a little bit longer. And the 3 middle ones are about the same, and the last one is long as well. But then the hind legs, if we check it here, bring it down just a bit. We've only got 4 on this one. So again, most things in this superfamily have have this tarsal formula, but not all of them. We looked at a couple last week that definitely did not have it. And I think today we will also look at some that do not have it. Except our first one here does. So that last one that we were looking at was the family Tenebrionidae, and those are the darkling beetles. This next one here is the family Melendriidae, and these are the false darkling beetles. So I will introduce this one specimen, and then we will talk about some of those characters. Ta-da! So this is Dicerca litura, which is one of the most common and by far like the nicest looking melendriad. They tend to be kind of plain overall, and they have a lot of structural diversity. So they don't change, but like there is a lot of diversity in what they look like. And so because of that, it is oftentimes very hard to characterize this family. If you check out that link that Tortilla just posted, that's to this species, but then you can click back out to like the whole family level, and a lot of them are not boldly patterned, and their overall shape is not super distinctive. So this is one that I have had a pretty tough time learning. Just again, because they have so many different looks to them, and they also like don't have a ton of distinctive characters that are unique to them. So when you get here in the key, it's like, oh yeah, it doesn't fit any of these other things, but what it does have varies a lot as well. So the first thing that we are going to look at and double check, remember we are in the Tenebrionidae, so we're going to look at feet. Don't be weird, chat. Let's see if I can get a good angle on either of these. That's not bad. Pickle. Okay. So we're pretty zoomed in on this one. Um, and so the field of view is not the best. What are we? This is a front leg. This is a front leg. How many are we counting? Four, four. What did I say it should be? Ah, space camp. He's got a little toe. Yes. So remember, tenebrianoids are supposed to have five. And this one, uh yeah, five. Exactly. First try, am I right? So this one, it has a fairly long first segment. And then this is two, three. This little lobe going under here is four. And then five is the one that has, again, the claws attached. So that little lobe is is different than what you see in the regular tenebrionids. Those typically have very uniform tarsi, kind of subcylindrical. Does not differ this much. So you're thinking about pseudo tetramaris, Molly, but this is not that. So in pseudo tetramaris tarsi, segment 3 is really big, and then 4 is very small. This one, 3 is kind of regular, 4 is small, but also big a little bit. So that's pseudo tetramaris. Pseudo is false, tetra is 4, and maris is number. But yes, this does not count as a pseudo tetramaris tarsi. Even though that fourth segment is reduced a little bit, it's not as reduced as we'll see later in the true things with pseudo tetramaris tarsi. Okay, so that was a front leg. Here's maybe a better view of that fourth segment. So how many we got here? There we go. We got the end in focus. Yes, space camp. Yes, yes, yes. 5. So one thing that you will notice about this one in particular, on the middle and on the hind legs, which is one of the things that is not fully diagnostic, but is one of the things that I will check when I think I may have a melandriad, is that first tarsal segment is super, super long. So right now we are looking at 1, 2, 3. The little lobe underneath is 4, and then 5 again has the claws. But that first segment is really, really, really long. And again, that is not fully like a dead giveaway, but it is something that I tend to look for. Okay, great. So 5, 5. Check a hind leg. can get all this in frame. Granny, what's up? So again, we've got that super, super, super long first segment. So this here is 1, 2. The little lobe segment there is 3. And then the end there with the claws on it is only 4. So great. This one does have the 554 tarsi that we're expecting of this subfamily. That's excellent. Finally able to catch a pre-Chatchats. Yeah, well, welcome in. Happy to have you here learning about ridiculous beetle ID. Do you guys remember what we were talking about with connate ventrites? Do you remember what that term means? On the topic of toes, do any bugs have an opposable thumb equivalent? No, not quite. Excuse me. The closest thing. I don't know if there's a good picture of this or not. It's kind of an obscure group. Oh, that's actually a really good picture of it. So we've talked about these a few times on regular chat chats. If you'll allow me to swap this over. So this is a hanging fly. This is in the order oh, I can't get on there. Okay. This is in the order Mecoptera, which are like the scorpion flies and scorpion fly relatives. And then this family in particular is called Betacidae. And again, they're called the hanging flies. And this guy is showing you exactly why they are called that. They will hang from their front legs. Middle legs are kind of just there. And then the hind legs dangle down. And if we come over to this Google image instead, you can see that there are these big grabby barbs on part of the tarsi, and there's only a singular claw here. And so what they do is they will, again, as the name suggests, hang with their tarsi down like this, and they will reverse praying mantis. Is the best way I can describe it, is that they will wait there until things fly by, and then they will grab them out of the air with their hind legs. And that's pretty crazy. That takes some pretty nuts coordination. But this is like the best example that I think could be like opposable, because you saw the way that that singular claw kind of curled back and held on to, um, they are able to hold things with their feet and they do articulate around to grasp things. So this is probably the closest thing I would say that there are in the insect world for um, opposable thumbs. But yeah, they dangle down and they grab stuff. It's pretty cool. So that's the best I got uh for opposable thumbs. Of course, Vic, anytime. How many pull-ups can he do? I don't know if he needs to do pull-ups. He just kind of hangs there. It's more of like a dead, a dead hanger, like that carnival game where you have to hold on for 100 seconds or whatever it is. So back to melon dryads. I was asking about connate vengtrites, and if we remembered that term, and the consensus kind of was not really. Roach, how's it going? Welcome to Pre-Chatchats. Connate vengtrites are the fused segments of the abdomen. A vengtrite is simply the segment on the bottom side of the abdomen. And so on the actual tenebrionids, so back in the real tenebrionids, the real darkling beetles, they will have 3 connate vengtrites. So if we grab our specimen back, maybe move this over a tad. Need to take these off. So this is sometimes a pretty tricky character to see, but thankfully on this one, it's actually not that bad. Connate, again, means fused. And because these are segments, there are intersegmental membranes that separate them and allow them to actually articulate and move their abdomens. If we look here, this is the first abdominal segment. And we know that because it is immediately behind the hind legs. All of the legs and wings on an insect are attached to the thorax. And then everything behind there is abdomen. So we've got our hind legs. This is abdominal segment 1, 2, 3. And then if we shift up just a bit, we also then have 4 and 5. If you can see this difference here, though, between 3 and 4, or no, 2 and 3, and 3 and 4, the membrane between 3 and 4 is a lot more visible. Let me just get it back here and focused in appropriately. So right here is the visible intersegmental membrane. And then up here, we can tell that it's fused because we aren't seeing that membrane. This can be a tricky thing to see depending on how the specimen dries. If the abdomen is really compressed or kind of tucked up, it can be really difficult to see those membranes. No gap. Yes. Courtney, hello. I think you were my 3rd streamer for the year. Maybe? No, 4th. Pickle Nick was 3. My goat. is what I'm saying. Um, but yes, so this is what I'm talking about with connate ventrites. And on true darkling beetles, there are three. So one, two, and three are connected. You had your last entomology class today, Sag. How did you do? Did you learn a lot? Did you know more than your professor? If you keep coming to chat chats, it's never really your last entomology class. Much learning. Excellent. Yes, that from experience. No, not really. When I was taking entomology classes, I did not know more than my professors. But I also didn't have a Twitch streamer that I was friends with that also had a PhD in entomology that I could talk to about things. Um, I've had other chatters though say that going into their entomology class, like, oh, I knew this from stream already. So I was just checking. I don't know how into intersegmental membranes and pseudotetrameris tarsi they get in your entomology class, but exciting. So back to now the melandriad or the false darkling beetle. It's not for everyone, granola. I think I got my time and experience out of it. So this is another one where we are going to be looking for connate ventrites and figuring out how many there are. And this one is definitely a bit tougher. That says differently. Like I said, not for everyone. Um, so let's relocate. Here are our hind legs. So then this is the divide between segments 1 and 2. Do we think this is fused or unfused? Looks stitched. That's not an option. So telling whether or not something is fused or not can be quite tricky if you don't have the other thing to compare to. So let's, for sake of learning, check down here and look at these last couple of segments. Yes, these are more open. So these last two are unfused. Oops. If we go back up, all three of those are kind of in view. They're more open. So yes, these have the same. This one in particularly has the same unfused fused unfused. No, that's not how it works. um, can that happen? I don't think that's like a pattern that exists, Granola. Um, the top one is a wider suture, but it is still fused. So this one has three connate ventrites, just like the true tenebrionids do. Now, this is my only specimen of this group, so I don't have the other one to show you. But there are things in this group that only will have two fused abdominal segments. And so again, it's keying these out. It's like, oh yeah, it doesn't really fit anything else, so we can kind of put it here. Only having two fused abdominal segments is low key kind of weird for this group. So this one not so bad, but I know other ones in this group have that odd character. And then if we come up here and we look at the face, so we're still underneath at this point. We'll turn a little bit more sideways, so we should use our cork instead. So we're looking at the side of the head. On the right-hand side, we've got the top of the pronotum. And what we can see from here is that the head is almost completely covered by the pronotum. It's sort of like a hood. Also, like tenebrionids, these guys have the antenna inserted under a little bit of a shelf. So you can see there is a slight ridge over this first antennal segment. Maybe. Love the shot. Cool. I'll keep that in mind for oh, this is maybe news. I think I said it on Monday. I am in the process of uploading the VODs from all of this. So if you've missed streams at any point from like early October until now, they will exist at some point. Tortilla, I think I fixed that thing that we were talking about. If you did, in fact, make the thing without being too... Yeah, there it is. We have a new command, everybody. Exclamation point YT for YouTube. I've got 3 up right now, and also a talk that I gave at my local Audubon Society at one point in Arizona. So those exist. I haven't had time to download the rest of them. The most recent one on there is I've got October 1st, 5th, and 6th. That doesn't feel... Oh, yeah, it does. That was a Sunday, Monday. Yes, bonus nerd shit if you are into that sort of thing. And like I said, I need to download everything from October 5th until now. So there will be a lot more going up. And I had to scrub through there and find interesting uh, thumbnail pictures. So I'll keep this one in mind for this video, Granola. So right here is what I was talking about. Is that this antenna is inserted under um, under this little shelf right here. But the thing that I wanted to talk about from this view is actually the shape of the palp. So the palps are this paired structure. Underneath, it's a mouth part. So there are two pairs of palps on every beetle. And in the case of melandriids, another thing that is, again, not fully characteristic of, but something that I look for with melandriids is the shape of this last palp. His punching bags. Yes and no, Shoveler. Or why did I call you Shoveler that time? Your last name, that's weird. Granola. Um, in spiders and arachnids specifically, they have these things that are called pedipalps, and those are derived from early derived legs. And they're the ones that form the little boxing gloves on males. In insects, they're not called pedipalps. They're just called palps. And there are actually two different sets. There is a maxillary palp, which is like the first set, and then the labial palps are generally much smaller. So right now, we are looking like the antenna is blurry, but then we have the maxillary palps here underneath. So it's a similar structure in that it helps deal with like food and keeping food in the mouth and things like that. But it is not the reproductive. It's not reproductive like it is on spiders. But good thing you're making that connection. It is roughly the same roughly the same structure. Okay, here we go, I think. This will be the good view. We're about to hit a new vocab term. So do we see how this one, I've changed the view again. You're still looking at the blurry antenna and then the palp there underneath. Do you see how it's like kind of narrowed towards the back, but then the front is super, super wide? This is what we would call securiform. Securiform, which means taco shaped. God, I wish. I wish that's what it was. Securiform generally means in the shape of an axe or a hatchet. I don't see it as that axy on this one. But there are other insect groups that have very securiform palps. Like this big ground beetle, for instance. This is like I said, a lesser example. When we are typically talking securiform palps, we are I need the foam. We're talking some shit like this. This one to me is much more axe shaped it is like this is full on just a triangle. um, the keys and things like that for melandryads also say that they have sacchariform palps. It is much less so on them, or at least on this specimen that we have right now. Some of the other genera in that group do have very broad triangles like this. So again, there's just a ton of variation in melandryads in these false darkling beetles that make them a real pain in the butt to identify. um, but yeah, so this is a good example of sacchariform. Um, keep that keep that tucked. There'll be a pop quiz at some point. But yeah, so there's that new term. And I don't really know if there's anything else big that I can tell you about melandryads. Aside from this one being pretty pretty neatly patterned. Um, just keep their food in their mouth. Yes. It's not like a lip, but it is kind of like they don't hold things with their hand or with their front legs to like keep it in front of their mouth. So they'll bite something and then they can use the palps to like support it while they're chewing. Um, yeah, it basically holds the food right here so they can actually eat with the mandibles. The palps don't do any of like the cutting or anything like that. They're simply there to like keep things right where they should be. It's like if you're holding a cob of corn like right here. That's what palps do. They're just like no, 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 no. And then yeah, now from the top, we can see that head again. And so we're looking straight down at the pronotum or that top part of the thorax, and you can barely see the head from above. Um, and that is, again, not completely unique to melandryads. There are certainly other groups that have that, and even some melandryads that don't have that. But yes, this is Dicercolatura. Kind of looks like leather. Yeah, so what you guys are seeing right now is what we would call micro sculpture, which is just like very faint. Ooh, do I have any in my pictures? Shoot, I don't think that I do. We might have to do that a different time. Um, but you're looking at like little faint impressions in uh, the pronotum and the elytra pattern, and they can be different. So there can be microsculpture on one and not the other, and the shape of them can of the microsculpture can be different. In ground beetles, at least, there are three main uh, patterns that you can get. Let me please hold. Figure. Aha. Oh no. Give me 1 second. I'm trying to track down my picture files for that book chapter that I'm working on, because I have a really nice picture of microsculpture. But I think I moved them all into my hard drive at work, so I'm just trying to see if I can access one of my files. Here, I'll give you guys the eventomology special of like the dancing fruit video for babies, which is just ants. Y'all like Calypso anyway. Okay. Look at ants for a second. Hmm. Okay. I apologize for removing the dancing fruits, but I did find what I was looking for. So if I switch to this, this is one of my plates, like I said, for that book that I'm working on. And this shows some of the different microsculpture patterns that ground beetles have. So on the left here, this one that sort of looks like chain link fence is what we call isodiametric. And so these are like little individual cells that are all roughly about the same size. They're all roughly hexagonal. And they form this really nice mesh here. Like I said, like a chain link fence. Then you have here in the middle, this is linear microsculpture. It looks like a bunch of deep lines kind of carved across the sides of it. Looks like fish scales. Yeah, it definitely does. Jalen, welcome in. Thanks for the follow and the first time chat. Hope you're enjoying the stream. And then over here on the right, we have elongate microsculpture. So this one is kind of a hybrid. The lines do tend to kind of mesh side to side, but they don't fully form those little cells like the isodiametric things do. And so sometimes identifying ground beetles comes down to the pattern and placement of this stuff. So it's like, oh yeah, head without microsculpture, meaning that it's like totally smooth. Elytra with isodiametric microsculpture, which would be that one over here. This one over here on the left-hand side. And so this is a pretty important thing to see, and it's also fairly difficult to see. The beetle that I took this linear microsculpture picture on was about 3 millimeters long. So it is unfortunately not as used on the larger ground beetles. Oftentimes, you're looking for microsculpture on very, very, very tiny beetles anyway. So, yes, wanted to pull that up. Let me swap back out of here over to the scope. and Rebecca. It looks like the microstructure of hair. Yeah, kind of. I can see that. Space Camp, I think, probably left to go to Amanda's stream, which is, whatever. But if she comes back complaining that there were no ants, y'all can tell her there were ants. She did, in fact, do that. Yeah, I thought so. It's okay. We won't cry about it. Okay. That was pretty good. Pretty in-depth there on Melangdryads. But now we are ready to move on to our next family, which I think I've talked about before at some point. I don't remember why I would have had these out before. But I distinctly remember talking about them. These are mordelids, or tumbling flower beetles. So, from the top, let's get in because it's a little small. Definitely smaller than our last one. So, kind of from above, this has a similar profile to what we were just looking at. The head kind of comes, or the pronotum rather, kind of comes up and hoods over the head just a little bit. And when we look at it from the side, you'll see that the head is pulled in to the pronotum a little bit, so it's slightly retracted. Do these? We can check it, but I'm just trying to see. Yeah. Okay. So these do also have 554 tarsi, but because this is kind of small, I doubt we'll be able to see it with this scope. But from the side now, you'll see a little bit different morphology going on compared to that last thing. The tarsi aren't actually that bad on this guy. So you see it is very humpbacked, and it's broader on the top than it is at the legs. So they're shaped like a wedge. So narrow on the bottom, wide on top. That means that they're basically always top heavy. And so the reason that they're called tumbling flower beetles is that when you get too close to their flower, they simply tumble off. If you're top heavy, you're shaped like a wedge, you can basically pull in all of your legs and you automatically tip over and will fall off of whatever leaf that you're sitting on. And I've had many students who have been seeing these on flowers, like actively sitting there watching them. And then they go to grab their aspirator, their little bug sucker upper that they have, and try to go for it. And then they just tumble off and you never see them again. So the strategy with catching these is sweep netting. So you take a net that's kind of thicker. It's almost got like a pillowcase texture to it. Excuse me. Because that way it doesn't rip as easily, and you will just go and lightly run the net over the top of leaves and other plants and things like that. And what you're doing is essentially taking advantage of this and many other insects' defense mechanism. Is that like, oh, my plant is getting bumped, let me get out of here. So they will either take off and fly or drop down and try not to get seen or stepped on or whatever. But if you're dropping and there's a net coming under you, you're going to get caught up in that net. So that's how sweep netting works, and that is typically how you end up catching these because you catch them while they're falling. They do fly, Jalen. Yes, they can. It's just not their go-to defense mechanism. It's not the first thing that they go to. They will oftentimes drop, and then once they land on the ground, they will then start flying. But if they're just going between plants, like there's nothing that's forcing them off the plant in any way, nothing disturbing it, they can fly from the plant just fine. It's just not normally how they go about things. The old tumble thing when they can fly. Yeah, definitely. Just a slightly different option. But some other things that you can see from this side view. One is the thing that makes Mordelids a Mordelid. And it is this elongate last abdominal tergite. So right here, this little spine that comes back. We were talking earlier about connate ventrites. Ventrite, like VEN, think ventral, is underneath. A tergite with a T is on the top. So the top of the abdomen are made up of tergites. The venter or underside of the abdomen is made up of ventrites. No, not a stinger. Not a stinger. Both males and females have this. And across the whole family, they all have this little elongate tergite on the top. I don't know what it's for. It's not like a springtail where Glacial, what's up? Welcome in. We're doing more pre-chat chats. Pointed and mostly long. Let me see if there's anything in here. I've never seen anything that like explains why they have this. What are we looking at today? So this one is a tumbling flower beetle. This is the family Mordelidae. I don't know if I had said that yet. Maybe I did. But they're wedge shaped, so they're very narrow on the bottom and kind of broader on the top. And their defense mechanism when they get disturbed is to basically tuck everything in and then roll off of a plant. But what I was just talking about was like the thing that I use to identify Mordelids. And it is this elongate last abdominal segment here that kind of makes this little spike. It's not a stinger. It doesn't... I don't know why they have it. I don't know if it like serves any specific purpose. But it is something that you see across the group. As far as I know. And always take that with some grain of salt because there are so many weird exceptions in entomology. But as far as I know, all Mordelids have that little spike. The shape of them is also very characteristic, so you might not need that spike to fully narrow you in. But then also, since this is the biggest Mordelid that I have on hand at least, um, that hind tarsus right here, below the weenie. I didn't pull that. It had to have been out when it died. But this one also has 554, so we can see a pretty clean 1, 2, 3, 4 segmented tarsus here on the back, which is correct for Tenebrionoidea. In theory, they should all have that, but as we covered last week, they don't all have that. You'll also notice that these antennae are quite a bit shorter than others we have seen so far. In many different groups, these are probably some of the smallest antenna that we've seen. The size of the antenna does not always equate to more segments. The kind of default standard for beetles to have is 11, and this one does in fact have 11 antennal segments. But most longhorn beetles, things that are known for their very long antennae, also usually only have 11. There are some groups where the segments are small and there's like 35 of them. But your average longhorn beetle will only have will only have 11 antennal segments. I think the reason that I just said 7 there is because my example I was going to use is that like humans and giraffes both have the same amount of vertebrae in their necks, but a giraffe is their vertebrae are obviously a lot longer. So sort of like this one, it has relatively small antenna, and even though it has the same number of segments that like a longhorn beetle would. So that's a Mordelid. Let's take a peek these other two, see what else I got in here. Other two are from Pennsylvania, so different habitats, probably different species. Just smaller to be cute. Yeah, so this one you can like really see how prominent that tergite is. Nice and extra pointy. Are you the same guy? Are you the same thing? Nope, you're different too. So again, that spike, pretty obvious, pretty easy to see. And then here are those two together. So you can kind of see some of the diversity and patterns of these guys. We'll go up from here. So the top one now is a little bit like orange and blacky. The bottom one is black mostly with some lighter gray patches. There is a really phenomenal looking one that I've never actually collected myself. It's a very large Mordelid. Large, for this group. It's not huge by any means. Hoshihananomia octopunctata. This guy. Let me see if there's a good picture of it. This thing. And so the ones that we're looking at right now are maybe 4 millimeters long. This one in the picture is like almost a centimeter. It's quite large. It's the largest mordelid we have in the U.S. But the pattern on that one is just ridiculous. And it's a widespread Eastern thing. I just haven't. I've just never encountered it. Doesn't look like there's any bug guide records on Florida, but I know they're also in the state. So I hopefully I'll find one soon-ish. If I do, I'll be sure to share them on stream. Okay. Not a ton to get into with that family. That one is pretty straightforward, all things considered. Um, at least in terms of identification, there's not a lot you can confuse these for, just because they've got such a distinctive look to them. Now we're moving into some weird territory. I'm gonna put this up here. I'm gonna let you guys consider for a second. Just really sit and think about this one. As it comes into focus. Da 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 da. What the hell do we got going on here? Again, I'll give you a second to take it in. Excellent question, Tortilla. Excellent question. This obviously does not strike you, or at least I wouldn't expect it to strike you right away as a beetle. So if you were to guess what this was prior to knowing that it was a beetle, what would you say? Fly with armor. Enraged kangaroo, wasp, weird fly. Okay, I can see both. I would personally lean wasp-ish myself. Um, but yeah, let's. God, where do we start? Okay. It's weird. It's a beetle. One of the things that we know that defines beetles is the presence of elytra, which this one does have. NFL shoulder pads. Well, I am starting with those. Um, so I can absolutely see why you would say fly. Um, because it does look like it only has two wings. These wings are quite exposed. They're very obvious compared to most other beetle wings. And that's because beetle wings are typically stored under these hardened wing covers that protect them, called elytra. Yes, like from Minecraft. Um, but these guys, as Tortilla said, had NFL shoulder pads. Um, yeah, so this little nubbin right here and this nubbin over here are actually the elytra. And they basically defeat the purpose of what elytra are for. Um, one of the hypothesized reasons that beetles are like so evolutionarily successful, meaning that they've got like so many species, is in part, we think, due to their development of elytra. What those hardened wing covers allow them to do is tuck up their flight wings that they actually need to fly and protect them while they are living in really crazy habitats. So if you think about it, there's really no way for a butterfly to live inside of a log or underground or in a really thorny plant because they have these big, delicate wings that if they get crunched and crumbled and cut and scraped and whatnot, they don't fly very well. With elytra, um, it it's basically like a sword sheath. It covers the sword when not in use. It can be very easy to unsheath it to get to those flight wings, and you don't lose the functionality. You can still fly, but you also now have the added benefit of protecting those flight wings so you can exploit different resource groups than other things. Butterflies were my example because they're obviously big and showy, but like dragonflies, even grasshoppers, they all have exposed wings. And you don't see any other group living in the diversity and the extremity of the diversity and extremity of these different habitats. Don't do anything so the communities don't live in small spaces. That's a good call, Jalen. Um, I will, however, say important note: they don't all lack elytra. So this is one that a friend of mine gave me from Bolivia, and this one has full wings. Wing dress under a elytra trench coat. Hell yeah, Tortilla. Well, you've got 10 months, 11 months. Start sewing. That's the same type of beetle. Yes. Yes, it is. Again, one of the frustrating high levels of diversity in beetles. Um, this one is just like so much less exciting. One thing that I will point out on this one, because it's a lot more obvious, or I guess less obvious, the body shape of this one is a lot more similar to the Mordelid that we were just looking at. Um, this, again, I don't think I've said this yet. This is the family Rhipiphoridae. No, not that. And these ones are called wedge-shaped beetles. So these ones get wedge-shaped, while Mordelids get tumbling flower, even though their body plans are relatively similar. These two the same family? Yes. So this one and the one that we were kind of looking at that we were like, oh, wasp-ish, maybe. But what I did want to point out with this one, in all reality, the more boring of the two, is the full elytra. But because they also share the name wedge-shaped beetle, if you look at them from the side, they're quite similar to the mordelids that we were just looking at. But there is no spine. Obviously, it's an insect. There's no spine anyway. Ew. But here on the end of the abdomen, that spine that we were just looking at with the mordelids isn't there. So even though otherwise they sort of look similar body shape wise, you know this one isn't a mordelid because it doesn't have that last spine on the abdomen. So we'll put this one back, and then we will go look at the cool one. Go back to our weird waspy guy. Does anyone have any ideas as to why this one looks like this? We'll unlock a new angle here and go basically straight down on these really crazy antennae. It's an athlete. Don't know what that means. Rarely land? Okay. That's a thought. Vibes only another species links to bug guide. Yeah, vibes. Tortilla's the goat. So we are now looking head on at this really weird beetle. And we have talked about antennal diversity on some of these streams before. What does having really big and really elaborate antennae allow you to do? Why do things tend to have really big, really elaborate, and really extravagant antennae? Smell better. Yes, exactly, Tortilla. So for those of you that aren't aware of this, insects don't have a nose like we do. They're still able to smell though, because they have these antennae that act as chemoreceptors, which basically means smell, or it's chemical coming in, but scents and smells and how things are perceived like that is all based on chemicals. So insects will use their antennae to smell and track things down. The bigger the nose is, hardy har har. The bigger the nose is, meaning that the more elaborate the antenna is and the more folds and bumps and hairs and things that there are on the antenna, it allows them to pick up on smaller amounts of certain chemicals and really be able to smell everything around them all at once to make informed decisions about where to go. Now, this is a male. Males tend to have the more elaborate antennae. And that's because male insects pretty much only care about one thing, and that is trying to track down females. So female rhipiphorids do not have as elaborate of antennae. And the males will use these super elaborate antennae to track down and try to uh, locate a female. Now, this one is a bit of a double edged sword. If anybody has read the bug guide link already, um, you've probably learned the cool fact about these is that these beetles are actually parasitic. So their larvae are called triungulans, which are these little roaming larvae that will get laid into the head of a flower. And I'm going to switch over here to my own bug guide page. Here we go. So right here in this photo, these little things back here on the flower, those are the beetle larvae of the ripiphorid. And what they're going to do is grab onto this bee somewhere. Under the abdomen, one of the legs, up here on the thorax. They will attach somewhere here to this bee. And then the bee will fly back to wherever its nest is. Because this leafcutter bee is picking up this pollen and picking up this nectar and stuff like that to take back to make their own nest for their own young. And so what happens is the legs of the ripiphorid, the eggs of the ripiphorid are laid in the flower. They then hatch. They then wait until a bee will show up like this, and then they will just grab on. Then they get taken back to wherever that bee had been planning to lay its nest, and then they hop off. And then they wait. And then they wait. And then they wait some more. And what they're waiting for is for that bee larva to get bigger. It grows to a certain size, then they go, here's my cue. I am going to burrow into this larva and essentially eat it from the inside out. So they lay their eggs on the flower, so the bee will be there. They will grab onto the bee, go back to the nest, and then eat that baby bee. That is their whole reproductive strategy. That is how they all work. They will send these little things out to crawl over and try to find bees. They will also do them on... there are some individuals, like some species in groups near here, that will feed on wood-boring beetle larvae. There are some that are on cockroaches, so it's not just beetles that get parasitized by this group. Definitely multiple species or multiple other insect groups that get parasitized by these guys. But so yeah, that kind of is why it looks more waspy, is because it's hanging around on flowers and it's using that to be like, yes, hello, I am here on this flower just like you, fellow wasp. Certainly no harm in landing here. And that allows the wasp to come in, the larvae to grab onto it, to feed, and then the whole cycle starts on over. So very cool antennae, very cool reduced wings, very cool life cycle in general. Um, so yeah, Rippifords are really neat, and they're relatively short lived as adults. Their whole thing is mate, produce eggs, and then just go. So they're available and like out flying depending on which species for a couple weeks of the year at most. And different species will have different timings of when they come out. So trying to track them down and find them is actually uh, kind of tricky sometimes. You need to know what type of uh, what type of prey they're going after. So if it's a cockroach one, or if it's a beetle one, or if it's whatever, you need to know that. And then seasonality, you should try and figure out that as well if you're going to try and like mount a collecting expedition for these specifically. But a very, very cool group overall. You don't see antennae like this on a beetle very often. And even not just the antenna, just like the whole thing is really odd. Again, going back to that, like, it sort of looks like a wasp. Very moth-like. Yeah, I think the antennae are quite moth-like. But the rest of this is, I would say, uh, wasp or fly for this thing. But anyway, that's that. Let's see if this one has. It's another cool thing about them as well, which we have seen this on other groups before too. But I think it's always kind of neat when we get something like this. If that'll, let's switch back to the more beetle-y looking one because they both have it, and this big one is going to be a lot easier to see this other thing that I want to look at right now. They got comb toes. So this is another one of those things that can help separate Rippifords from the Mordelids. Mordelids, again, as far as I am aware, none of them have, we call this pectinate or combed tarsal claws like this. In Rippifords, I think they all do. Uh, Again, there are always weird exceptions to things. I don't know if that is totally accurate across the board, but at least I think the U.S. Rippifords all have comb feet like this. So the Rippiford eats the bees and then grows up and reproduces near a new bee. Yes, that is the gist of it. The beetle larva will feed on the bee larva, go through its whole life stage. So it'll pupate, go into the little cocoon, or go into the little pupal chamber, and then emerge as the adult beetle. And then it will come out and then go to a flower patch. The females will go to the flowers. The males will use their crazy antennae to locate the females that are on the flowers. They will go there and mate, lay their eggs in the flowers. The larvae will hatch, hitchhike on the bee, go back and eat the bee larva, and so on. So yes, that is indeed how that cycle works. Yeah, we got time. We got time for one more. Like always, I'm going to take a little bit of a break between this and regular chat chats, probably here in about 15 minutes. Um, we're going to go over one more family, and I think you guys will like this one, because I think it's one that you're probably at least slightly aware of. So if we bring this back in, we're going to have to zoom out a lot. This is another big one. Their wings aren't protected because they don't need a long adult life cycle. I think that's probably part of it, Jalen. It also lends to their more bee or wasp like look to have the uncovered wings. Um, but yeah, that's probably how that ended up happening. That's a weird beetle. The one that we're looking at now, or the one that we were just looking at before? This guy's made of black licorice. Is that a good thing, Tortilla? I kind of like black licorice. Didn't see the one before. It's a freak. You'll have to check the VOD on this one when it goes up. Not on the YouTube, but like when it just is like available on Twitch. Yeah, go back and watch this last one. It was a weird looking dude. I think everyone that was here can probably attest to that. Huge fan. Okay, cool. Based. Um, this. Does anybody have any guesses before we go? Before I tell you at least what this one is. Bug. Okay, not wrong. Not wrong. This is a family that you've heard of. Beetle. Yes. Okay, great. We're getting somewhere. Clamter, beetlebug, carabid. No, not a carabid. We've covered carabids already. This one is a zoopharid, which are the ironclad beetles. So these are the ones that are like extremely, extremely tough and hard with their exoskeletons. This is not the one. So this is not Phleodes diabolicus, which is the diabolical ironclad beetle. And the cool thing about that one is that Phleodes diabolicus has one of, if not the hardest exoskeleton of any insect. And they did a study on how the elytra and the rest of the exoskeleton was constructed. And they basically figured out that the inside of the elytra structure is basically made like Lego tiles. So you'd have two pieces that come together, and then on top of it would have another piece connecting them. So there's a bunch of little pieces that are sandwiched and layered and everything like that. So it makes the elytra really, 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 really tough. And they didn't do this for the experiment, and I want to be very clear about that. But they wanted to test how rigid the exoskeleton was. And so they ended up applying, they had like a force meter that would push down on the elytra. Um, and essentially, what happened is that the amount of pressure that it took to actually like crunch this beetle from the top, like pushing straight down on the top of the abdomen, um, was higher than if you were to drive a car over it. So just think about that. An insect that could theoretically survive getting run over by a car just because it has this super, super tough exoskeleton. Gotta be so annoying to pin. Let me tell you, Glacial. Correct. Um, are these bark beetles? Sort of. This one in particular, this is Zophorus nodulosus. No. No, it's not. Uh, hold on. They moved things around. This might not even be Zophorus anymore. No, no, it is. Okay. This is Zophorus gracilis. Liar. Just got my species mixed up. Um, so these ones are up on pine bark in Arizona, or at least out west in general. You find them at night on pine trees, but not all ironclads are bark beetles. Zophorus nodulosus, the one that I was just confusing it for, is the big, like, black and white uh, ironclad beetle. I have bug guide. Oh, Tortilla's got it. Beat me to it. Love. Oh, Gracilis. The one above was the one that I misspoke. But yes, so these have ridiculously hard exoskeletons, and these ones aren't. This one in particular is not terrible. Um, I did have a couple of specimens of the big black and white ones from Texas, and it took me 3 pins to get through the specimen. One to get through the top of the body, one to get through the bottom of the body, and then 1 pin to actually keep the specimen on. So it was a whole process, and I was like, how do people deal with this? Like, this is not a good way to pin specimens. And so I learned 2 things that day, because I posted it on Twitter at the time. But I learned a neat little way to work on it. So you take your forceps. The end that you actually pick things up with is down on this side. You flip them around. So the part that actually closes like down in your hand. Then you take your pin and you slide it inside the forcep. So you have this going on. And you slide it up as far in there as you can get. And now you have a lot more leverage to actually be able to pin down through them. And that's how I was able to do this one that we're currently looking at. This is not the hardest of darkling or of ironclad beetles. But then the other thing that I did learn for folks that work on the big ones in the tropics, like 1.5 inch long tropical zopharids, you do actually use a drill. I know people were memeing about that. Tortilla said that, but that is like legitimately how they do it. They have a drill press, so like one of those things where you can lower the drill down specifically, that are made for watchmakers. So it's a very, very fine drill bit. It's very small. You're not taking like a 3/16 like and doing it that way. But you do have a jeweler's drill press or a jeweler's drill to make the holes through the beetle, and then you just put the pin in, which is a whole thing. The poor man's version of that is using the pin and the forceps option. But what I will also do is you can take a pin that is slightly larger than what you're going to use. So, like, if I plan on pinning this with a 2, I'll take a 3, and I will put the pin where I want it to go through, and then I'll sit there and just spin it back and forth. So in my hand, it's making a little bit of a drill, and so it just thins out enough of that exoskeleton that I can actually get the pin through normally. Um, Pilot holes. Yes, exactly. Exactly, granola. So there's a couple of ways to deal with these really big, really tough exoskeleton beetles if you're going to be pinning them in this way. You can use the drill. You can use the backwards forcep thing, which is my favorite. You can use the little finger drill with the pin. But yes, those are your options. Are beetles the toughest bugs? Yes. Phleodes diabolicus, the diabolical ironclad beetle, the one we were just talking about, is, as far as we know, the hardest exoskeleton insect on the planet. Velvet ants, for whatever reason, the family Mutility, like those big red wingless wasp things that look like ants, those also have a particularly hard exoskeleton, but nowhere close to the level of ironclad beetles. So, how do we know that this one's an ironclad, right? Superficially, from the top, this does look a lot like a regular darkling beetle. Again, it's the same superfamily, so it makes sense that they look somewhat similar. But the key to it, we're going to have to flip them over. And so, first of all, this is a cool thing about this species in particular, is that it has these very dense lines of yellow setae going underneath the femur and the tibia. So there's this really nice like section of golden hairs, which I think is cool. Ta-da. But then the other thing that you're looking for, we're going back to checking out abdominal segments. So with cool flocking, yes, it does look like flocking. So on regular darkling beetles, like we looked at the beginning of stream, they only have 3 connate vengtrites, right? They only have those first 3 that are connected to one another. Ironclads have 4. So the first 4 abdominal segments are essentially fused together. So if we take a look at that, this here is 1, and this here is 2. So we have a really deep line here, but it's completely solid. There's no like membrane that we're able to see between those 2. And then you've got segments 2 and 3. again, fused right there along that whole area. And in regular tenebrionids, that's where we would stop. That's where we would stop seeing that fused segmentation. Here, with the ironclad beetle, we actually have one more fused segment, and that's here between 3 and 4. And then between 4 and 5, you can see that intersegmental membrane has come out to play again. We're able to see a little bit in there of where the articulating abdominal segment is. There are other differences that go on here. There is one character that is about the distance between the hind coccyx in relation to the width of the middle coxa, which is like a weird length width ratio thing. Because it's science, is it significantly harder to get their genitals out than other beetles? I don't think so. Because when you go in for genitalia, you are opening the back of the abdomen. You're not really going through the exoskeleton at all. So here, this is the tip of the abdomen, like where it would open up. So you would just open this here and go in with the forceps and then be able to grab it out. I don't think I've ever actually dissected one of these. I don't know that I've needed genitalia on one of these guys, but I would imagine that it's not that bad. Because unless you are doing like the really in-depth type of dissection where you take off the whole abdomen and then basically grab the genitalia from the top rather than from the end, you don't go through the exoskeleton. You just kind of open them up and then take it out. Now, this is like the typical ironclad beetle that most people think of with this group. Like the rather large, dark, very hard to pin beetles. However, as with other groups, there has been some somewhat recent classification changes. And so now, indeed, this guy this is Bitoma quadrigatata. This guy is now a zoophorid as well. So this thing and the last thing are the same family. I feel like there's netwing beetles, what they're called. Yeah, sort of. I can see the resemblance there. So this is a colidiine or a colidiine. They used to be their own separate family, Colidiidae. They were called like, I actually don't know. Type it again. That's fine. They're called cylindrical bark beetles. That's what it is. So yeah, these used to be their own family, but now they are within the ironclad beetles, and that was a pretty big change when it happened. But it has all of the same characteristics that we use to define ironclad beetles. So if we were to flip this one over, it would have those 4 vengtrites that were glued or connected to each other. It would have the cocci that are very close together compared to tenebrionids where they're further apart. And it shares the 554 tarsal formula of like all of the tenebrian noids. So fun little one to round out the night here. Cool red spots on it. I did see another question that I missed. Granola. When would you need to know genitalia? More often than I would like to admit is the real answer there. Um, in ground beetles, which is the group that I primarily work on, you need to know genitalia a lot. There are a lot of species that look very, very similar to one another. And externally, you're like, yeah, there are a couple of little differences here that we can see, but it's hard to like actually be sure about it. If you do a dissection, you can get pretty sure pretty quickly. And the reason for that is that insect genitalia is often described as like a lock and key mechanism. Meaning that, like, imagine taking your house key and taking your car key. Your house key is not going to start your car, and your car key is not going to open the front door of your house. And so, biologically, what that means is if your genitalia is a certain shape, you're not going to be able to mate with a female with different shaped genitalia than yours. So you have to find the correct male genitalia that matches with the correct female genitalia. And then you're successfully able to mate. And looking at genitalia is a way that we can actually try to figure out which things are separate species and which ones aren't based on reproductive capability. With insects, a lot of the studying that you do with them is based on dead, dried specimens, like what we've been looking at all night. So you can't really tell, like, oh yeah, these two, I saw them mating with each other, right? Because they were probably collected 50 years apart. If you do genitalia dissections and there's not like a complementary structure in the males versus the females, it's very likely that those two things would then be different species and not be able to mate. Do they try? Yes, certainly they do. I have seen males of two different beetle families trying to get it on. And if you put close enough relatives in with each other, oftentimes you can get hybrids, but we don't know how often that happens in nature and how viable they are, and it's a whole separate thing. So, like, you can, in theory, try. But looking at genitalia is a really big thing for taxonomy, again, because you can't see them mating in the wild because they've been collected in the collection. So you can only use what you have available to you to basically try and determine, yeah, these probably could or these probably couldn't mate. Male genitalia historically has been a lot more focused on. Sexism in science, whole thing. But more recently, there's been more coming out and more using female genitalia for separating species out and things like that. Just because it's, roughly 50% of the population. If you have a thing that you're trying to identify and it's female, and the only way that you can identify it is with male genitalia, what are you going to do about that? Like, there's nothing you can do about that, and it's a real shame. So nowadays, more folks are doing that. And then yeah, if you find a male, great. If not, oh well. Oops. But great. We got through 4 families tonight, which I think is pretty good in about 2 hours, maybe half an hour or so per family. I am. Thank you, Luna. We get festive around these parts. Um, I am going to send you guys to Amanda first, and then Amanda will send you to Alveus for chat chats. So in about 20 minutes, we are going to be doing the we're going to be using the cameras at Alveus Sanctuary to go around and look for bugs and also talk about whatever bug topics chat wants to go on about. So feel free to stop there. Going to work soon, my chat chats, but have fun. Night, Roach. Hope you have a good night at work. Amanda Express. Is that correct? Yes. All right. I'm going to send you guys over there. Follow Amanda. If you don't, she's also Alveus staff. And then when she's done, she will raid into Alveus for bug hunt. So I'll see you guys in a little bit. Bye.